Hello everyone. Today we will discuss about the topic gonorrhea. The gonorrhea is classified into three types. The first type is the gonorrhea which causes the infection in the urinogenital tract. The second one is the extragenital gonorrhea which causes the infection in the mouth, rectum, pharynx, tonsil and the eyes region. So third one is a metastatic which causes the infection in a severe cases, means it leads to the complication. Morphology. So it's a gram negative bacteria, non-sporulating. It doesn't form any spores. It's a non-motile aerobic bacteria, and also it's a non-acid fast bacteria, diplococci in shape and kidney shaped. Cultural characteristic. Media used to culture the gonorrhea is the chocolate agar media or the Thayer Martin media. In Thayer Martin media, it consists of the vancomycin, colistin, and the mystine, which is suitable for the growth of gonorrhea. The pH provided for the suitable growth is 7.2 to 7.6, that is neutral pH, and the temperature is also optimum temperature, which is 35 degrees Celsius. So when we grow the four types of the colonies, we can see when we culture this gonorrhea. So the first two types will be brown colored, small, pilated and the virulent forms, and the third and fourth type will be large non-pilated virulent forms. So the biochemical reaction where it is a catalase positive, means where bacteria consist of enzyme which converts the hydrogen peroxide to the water and the oxygen gas, and it's a oxidase positive where the bacteria uses the oxygen as a final electron acceptor in the respiration process, and it's a indole negative where bacteria can't convert the tryptophan to indole when the indole test is done. Antigenic property where the pilus and the lipopolysaccharide. So pilus we know that, and lipopolysaccharide it is present in the cell wall of the bacteria. So this both pilus and lipopolysaccharide when it helps in adhesion, so it acts as a antigen. Resistance, they can be killed, the bacteria can be killed if we continuously keep it under the heat for 2 hours, and it is highly susceptible to antibiotics like penicillin, tetracycline, ciprofloxacin, etc., that I'll discuss later, and they can be killed by treating with a sulfonamide also. So pathogenicity, which causes the venereal disease and can spread through the sexual contact. Adhesion through villi lead to the sterility of the male or female. So it causes the gonorrhea. It affects the both male and female. How it is going to affect means the first step in this is adhesion, where the pili acts as a antigen and go into the host. After the micturition, means after the passage of the urine, it passes through the intracellular space of the connective tissues. So in the male, it in acute urethritis, mucus discharge containing the gonococci spread from the urethra and to the prostate gland, and then to the seminal vesicle, epididymis. So in this way it affects the male. In case of the female, in the urethra, cervix, it goes through the urethra, cervix, uteri and the Bartholin glands, endometrium and the fallopian tube. This leads to the sterility of the female when it reaches to the fallopian tube. So also it causes the arthritis, meningitis or gonococcal ophthalmia. Gonococcal ophthalmia, it is seen mainly in the children's, means the newborn baby, where the during the passage through the birth canal, it affect the eyes of the newborn if the bacteria is present in the birth canal. So the laboratory diagnosis is done in two stages, that is acute stage and the chronic stage. Acute stage, it is easy to diagnosis where the sample is collected from the in case of the female we collect the sample from the cervical swab, or else in the male, in case of the male, the urethral discharge is collected as a sample. So the cotton is dipped in a saline water before. So that is the salt water. We dip the cotton in the saline water, and then we are going to collect the sample and culture that sample and in the chocolate agar media with a 5 to 10% of the carbon dioxide with the optimum pH and the temperature. Temperature should be 35 degrees Celsius to 36 degrees Celsius. So after the culture is done, the loop full of culture is collected and we prepare a smear on the clean grease free slide and observe under the microscope. But in case of the cornic, the morning drop is examined after. So how we collect is the morning drop is we have to massage the prostate gland, and then we have to take the sample out and the smear is prepared. So before the smear we have to culture, right? So the Thayer Martin media is used particularly during the cornic stage. So after culturing in the Thayer Martin media, so we have to prepare a smear and use the fluorescence antibody technique. So treatment, the gonorrhea can be treated with the antibiotic like penicillin, tetracycline, ciprofloxacin, aflatoxin, etc., and also with the sulfonamide. Prophylaxis, there is no availability of the vaccine. Health education is very much important, and the contact tracing is also important, and we should avoid the sex with the multiple partners and the infected person. So if you like the video, please hit a like button, share with your friends and subscribe.